Let's start with a review question. Which of the following is not a reason why antiviral agents are hard to develop? Is it A, because viral genomes are so small, there are relatively few molecules that can be used as drug targets? Is it B, antiviral agents usually have severe side effects because viral replication involves many host cell processes? Is it C, to destroy the virus, the host cell must also be destroyed? Is it D, drug-resistant strains develop rapidly because viruses mutate quickly within the host? Go ahead and pause the video. Think about this one. The answer would be C. To destroy a virus, you do not have to also destroy the host cell. Sometimes our immune system does do this, but this is not the only option. Antiviral agents often prevent viruses from replicating. They might impact cells in the process, so they do have severe side effects, but they do not, by definition, have to destroy the cell that contains it. This is true, right? So C is the answer. It's not a reason. D is true. Drug-resistant strains evolve rapidly. A is also true. Viral genomes are small, so there's very little to target. And as we said, B is definitely 100% true. There are a lot of side effects there. In 12.2, we're going to talk a bit about viral structure and diversity a bit. We'll talk about different viral genome sizes and what makes up a virus, and talk about why they evolve so quickly. So a virus is relatively simple. It has a genome that's either made of DNA or RNA. There is a capsid, which is a set of proteins that surrounds and protects the genome. So here in this influenza virus, this is the capsid. Some viruses create an envelope which uses host cell membrane around them. So you can see that here, this green bit. And then there are a limited number of other proteins that might be associated, like these spike proteins, which help the virus get into the cell. Something like a phage, it has a capsid. Its genome is contained inside of the capsid. It might have this little neck and tail and these tail fibers that help it inject its genetic material into a bacterial cell. So very small amount of components there. Viral capsids often form what is called an icosahedron, which is a 20-sided polyhedron with little triangle faces. So if you're a D&D player, you probably know the icosahedral die is your D20. This is herpes simplex virus, for example. And this shape is formed because this geometric construction happens when you have specific protein shapes. Viruses are all about small, compact genomes, so they repeat this protein over and over again, and it forms these triangular faces that arrange themselves this way. So this is often the shape that we get because of the limited number of proteins that are encoded in the viral genome. Like we said, some viruses, like herpes viruses, also encapsulate themselves in an envelope. This is taking host membrane. So it could be from the nucleus, endoplasmic reticulum, or the cell membrane. And as they come out of the cell, they are coated in host cell membrane. Now, think about what that's going to do. That's going to essentially hide the virus, right? Because on the outside, it looks like a human cell, or it has membrane from a human cell. And the immune system is not going to recognize that as well. The envelope generally will have spike proteins that are in it, and they often are involved in binding to receptors on the surface of cells and getting the viruses into the cells. Phages have this kind of lunar lander structure. They have that icosahedral head capsid there. Inside of that is their genetic material. The tail structure is really interesting. This helps inject the genetic material into the bacteria. And in this example, we have T4 phage. It's landed on an E. coli cell. So you can see this is an E. coli cell, and this is the virus. It's way smaller. The virus is actually under pressure. So this genetic material in here is under pressure. And when it punctures the cell, it shoots the genetic material into the cell. So it's a cool little structure. Nowhere in here do we have a cell membrane, right? So viruses are certainly not living. They have no cell membrane, which is one of our prerequisites for being alive. The genomes themselves can vary in size, but they are generally small. And when I say small, they are like fractions of a bacterial genome, which is fractions of what a human genome is, or something like that. RNA viruses tend to have the smallest genomes. They just have a couple of genes. 1, 2, 3, 4, 5, 6, 7, 8, 9 genes in this 7,000 base pair RNA viral genome. These are just like the bare minimum genes. They rely on the host cell to provide all the other genes. RNA viruses tend to have higher mutation rates because of the processes they go through. RNA is less stable than DNA, and so in its replication, more mistakes get made. There's also not proofreading steps a lot of times in creating RNA. Retroviruses, the ones that go from RNA to DNA, that reverse transcriptase process, it's very error prone, so that increases the mutation rate as well. DNA viruses tend to be larger, but they're still small. This is 150,000 base pairs, which is about the size of a plasmid that we would see. A bacterial genome is about 5 million base pairs. So there's all kinds of different proteins that are encoded in here. There's much more complicated regulation that can go on. This is a genome of herpes simplex virus, which can lay dormant in human cells for long periods of time. Viral evolution is subject to the same forces as everything else, that even though it's not living, it can evolve because it's made of genetic material. We have this term antigenic drift. This is rapid mutation, so the accumulation of mutations over time. This happens rapidly because so many viral particles get made so quickly that mutations accumulate quite quickly in viruses. Antigenic drift is one process. We will talk about antigenic shift in a later section here. Every time a virus is replicated, it has the possibility to have errors in replication. This is going to introduce mutations and allow it to evolve. This is why the best way to prevent something like the COVID virus from evolving into different forms, right? We've had all these variants. The best way to do that is to keep it out of people. So don't get sick with the virus. Every time the virus enters a person, it has the possibility to replicate and in that process to accumulate mutations. When it's outside the cell, no replication occurs, no chance for it to get mutations. When mutations happen, they're subject to natural selection, so whatever allows the virus to reproduce better is going to be selected for. This often means things like changes in the spike protein that change, like here we have triangle spike proteins. If they change to squares, the immune system may no longer recognize those. This is why each year we have to get a different flu vaccine because influenza goes through rapid evolution. It's an RNA virus. So new variants are constantly evolving. Viruses evolve at several different levels. We can think of them as at the community level, which means evolving to infect different species. So something like there is a equine herpes virus, which infects horses. There's also a murine herpes virus in mice. They evolved from a similar strain, but they evolved differences to infect different species. Within a species, there is a population of viruses that will evolve. Strains will evolve that have different infectivity, the ability to get in, and virulence. So we've seen this with our COVID, right? We saw the Delta variant and the Delta BA2 or whatever it was, Omicron, things like this. So strains will constantly evolve within a species population. This can lead to different strains of viruses evolving that infect different places. There are different herpes simplex viruses, herpes simplex 1 and herpes simplex 2. One of those is an STD. One of those infects other tissues. We have varicella zoster, which causes chicken pox, which is the red itchy rash. We'll talk about that later in the course. I realize now, after teaching this class, that many of you may not have experienced the chickenpox if you're young enough because they developed a vaccine for it. So most of you have not had to go through the pain of having chickenpox. But if you're of a certain age, you certainly have gone through that. And you may remember chickenpox parties where one kid would get it in the neighborhood and everybody would go around and play with them to get exposed to it, so they would be exposed to it, essentially vaccinating them against other diseases like shingles. Varicella zoster is closely related to cytomegalovirus, which does not really cause the lesions and things that chickenpox is known for. So a related virus, but different way it affects us. And then there's viral populations within an individual organism. So a person, right, inside of me, viruses would evolve different forms, right? Variants might evolve that are resistant to therapeutic agents. Hepatitis C and HIV are well known for this. They evolve into diverse strains that can infect different tissues within the host and will become resistant to therapies. HIV is a case study in evolution. You give someone drugs to treat their HIV, it will evolve and it stays with them for the rest of their life, and that evolution will continue to occur. So this is just, again, an overview of the subject. It's so deep, it's fascinating, but that's as deep as we're going to go. We have viruses. They consist of a capsid, which is protein subunits that encapsulate basically their genetic material. The genomes can be large or small. RNA genomes tend to be small, whereas double-stranded DNA viral ones can be large. There are some viruses that have an envelope that's derived from host membrane tissue. Then it's going to have spike proteins in there. And then viruses are constantly evolving, just like every other biological organism. They are not alive, but they do evolve because they have genetic material. This is the weirdness of it, right? They're going to evolve to respond to selective pressures, so things like the immune system or host range or therapeutic drugs. They're going to evolve to adapt to those pressures. All right, that's it for 12.2.